Hi Toby. So I was with the Soil Mentor group on Wednesday, and we were having a look at your field that I think had previously been arable, but you now had in a herbal ley. And I think I believe you said it was the second year in herbal ley. So I couldn't resist but take a wee sample of the soil back home, just to have a look at the microbiology under the microscope. So I thought I would send you a video. Just I thought you might find this interesting. Um, so very briefly, here is the soil right now, and this is times 400 total magnification, so times 400 what you can see with the naked eye. And I was kind of suspecting that it might be incredibly bacterially dominated. I know you said that previous treatments had involved glyphosate, Roundup. I should imagine if you were farming it conventionally, as it were, there would be inorganic nitrogen and various fertilizers added, and it was probably ploughed as well. So this really, as I'm sure you know, really knocks back the microbiology, both the diversity and the populations and the trophic levels. So what we find in fields that have been treated, in conventional ag, or what I've kind of found like all over Scotland, many of our agricultural fields are entirely bacterially dominated. So that means that large populations of bacteria, but actually very little diversity within the phylum. So yeah, we're just having a look at the shapes. We want to see as many different shapes as possible. So I'll just explain a bit what we're looking at here. So as I say, 400 times total magnification. Everything that you can see that's gyrating around like this are bacteria. So we've got multiple layers of bacteria as I zoom in and out of the sample. You can see all that gyrating around, and they're actually moving around through a Brownian motion. Some of them have flagellum and a moving of their own motive force, but a lot of them, something to do with thermodynamics and atoms, are actually jiggling around, but I don't understand how that works exactly. But we've got quite a lot of bacteria, I would say. We do indeed, just on my first little scan here, it does indeed look like there's generally quite low diversity. We're wanting to see different types of cocci, so those are the little full stops that you can see. If I zoom in and out here, so just round little balls. We want to see bacillus, so those are kind of the more classic rod-shaped bacteria that most people are familiar with, and not seeing many of those. It's quite good to see lactobacillus. They can sometimes be a slight indicator of anaerobic conditions. But like I say, we're really aiming for diversity here. So this little guy here that looks man-made is in fact a diatom. There you go. So that's a photosynthesizing organism that makes its outer shell out of silica. You can tell that it's dead. So this diatomaceous earth is made up of trillions upon trillions of these animals, and it's no longer got anything happening inside. The organism has been killed from whatever reason. It's no longer photosynthesizing because it would be green in appearance then. But because the shell that it creates is so recalcitrant, it's just going to sit in the soil for a very long time before it breaks down. The other thing worth noting is there's a lot of mineralization in this soil. So there's not a huge amount of structure. I think I took the sample from maybe half a foot down or something, which is a wee bit below the rhizosphere. And it was also where the sort of worst aggregation was. There wasn't very much aggregation. It had a kind of loamy like texture to it, actually quite nice, but it didn't have that cottage cheese kind of little bubbly texture to it that we're looking for. And I'm sure you know all this, but all of this structure is created by microbiology. Generally speaking, bacteria are creating the smaller of the microaggregates, and the fungi create the larger of the microaggregates. So yes, you can see that it's all quite a mishmash of minerals. So for example, like this, I'll just put everything that I'm talking about in the very center of the screen. that little transparent plate there in the center, that's a bit of silica. So that's a bit of sand. The lighter honey-coloured little aggregates that you see spread around, they're actually fulvic and ulmic acids. So that's a good sign. That is a sign that there's definitely been historic fungal activity. Fulvic, or at least my understanding of it, although this is a bit contested at the moment, my understanding of it is that fulvic and humic acids are created by fungal activity, the breaking up, the digestion and the excretion of organic matter, and it's constantly complexed every time it goes through a microorganism's body, and then eventually we end up with humic acids, which give that color to the soil that we were after, that nice dark color. But yes, a lot of fulvic acids, and fulvic acids I think are generally produced by some of the younger orders of fungi. So the various phylum of fungi that haven't been living on the earth for as long as some of the Basidiomycetes have been. So what we're doing here in a healthy soil sample, and this is applicable to agricultural fields, we're looking to see at least a fungal hyphae per field of view at this kind of magnification. So you can see we're seriously lacking in fungal activity. The other thing that's worth noting is I haven't seen a single fungal spore yet. So it's my guess there's been really quite a long time since any fungi have been active in this field, because it's quite common to see, fungi can go into survival mode and asexually produce spores if they need to, which can sit dormant in soil for a very, very, very long time, but I'm not seeing any. I mean, thousands of years potentially. So, what else we got going on in here? The other thing that we're looking for is all the predators. Here's another diatom just here, or a bit of a diatom. The other thing that we're looking for is the trophic levels of predators. So there's the protists, basically, nematodes, encotriads, earthworms actually would be regarded as predators. Their main food is actually bacteria, protists, fungal spores, anything that's living on organic matter. As they're consuming organic matter, they're actually getting all of their nutrition from the microbiology that's living on that. So they are kind of predators. So these predators will excrete excess nutrients from their prey as they consume them. And they'll be doing this, of course, around the rhizosphere. And that's what gives plants their nutrition. So it's actually one of the only ways that plants can get their nutrition is from this predator-prey relationship. So yeah, a lot of bacteria. It's rod-shaped bacteria there. So yeah, I'm not seeing, we're missing a lot here, unfortunately. Of course, this isn't necessarily representative of your whole field. We just dug a 25 centimeter by 25 centimeter square right in the middle of it. So my suggestion would actually be we're at a point now in this field, and I'm not speaking as a consultant here, of course, but my best suggestion would actually be to make some extremely fungally dominated compost, because there's not even any fungal spores to germinate that I can see in this soil. So I suggest that we're really needing to reintroduce and inoculate this soil with a lot of microbiology in order for you guys to reap all the benefits that brings, in terms of plant health and animal health and human health and all that. So yeah, huge numbers of bacteria. So yeah, we really need these predators to control their numbers a wee bit and make some plant available nutrients. Let's just have a wee zoom out, see if we can spot any nematodes, any of the larger species that dwell in the soil, any mites, anything like this. They're very easy to spot, they're wriggling around, so we'll have a wee look for those. But the potential for that soil, I mean, it could be absolutely incredible soil, I think. So next time I'm up, I'm quite happy to take a few more samples back home, and I can have a look at fungal to bacterial ratios for you. What might be nice actually is to do a comparison of what you reckon your best fields, your most healthy fields to be, and we can do a microbiology comparison to this field, and that would be quite nice. I think it would be quite a stark contrast, I should imagine. So yeah, just in summary, definitely need more fungi, definitely need more of the predators, the trophic levels of predators, and they will naturally recolonize the soil, but it will take them a long time because they'll be coming, I suspect, from your hedgerows. Although if there has been quite a lot of spraying and various applications going on in the fields, it's not necessarily guaranteed that the hedgerow soil will be in good nick. But yeah, it's really exciting because it's good to get some baselines, and it will improve dramatically if you can find a way to add some microbiology to the soil. No plastics though. I'm not seeing many plastics, so that's quite good. Finding more and more microplastics in soil at the moment, but it looks like you managed to escape that, which is good.